All right, before we begin, are there any questions? Sorry for being a little late. The I had to log out of my Canvas account and reload the page, and that for some reason Zoom would not start with the page I had open, so I had to log out and reload the page. All right, no questions. Let's begin. Let me share my screen here. There, you should be seeing my screen now. Oh, good. The lecture has finished so that my computer won't be working so slowly. Today is February 5th, and we should be discussing Lab 6 today. Remember last time we did discuss the unknown project. I will be emailing you results probably late Tuesday night, as well as your unknown number. And it might be very early Wednesday morning, but late Tuesday is what my goal is. All right, Lab 6, diffusion, evaluation of disinfectants and antiseptics, and antibiotic effectiveness. There is some reading to do before this lab, so please read about the terminology of microbial control on page 177. Read about the principles of effective disinfection, evaluating a disinfectant and types, page 187 to 194. Read about the chemical methods of microbial control from page 196 to 197 and table 7-8. Read about antibacterial drugs, antibiotics, page, what's that really just page 1554 to 555. Read about the test to guide chemotherapy, the diffusion method, page 567 to page 569. There will be a virtual lab today, as well as video clips. The learning objectives: 1, be able to identify the type of chemical agent and the active ingredient used in various disinfectants or antiseptics. 2, evaluate the effectiveness of disinfectants and antiseptics using the diffusion method. 3, determine the mode of action of various antibiotics. 4, evaluate the effectiveness of various antibiotics using the diffusion method. 5, list the variables that influence antibiotic diffusion susceptibility test. And 6, be able to define the terms. What's sterilization, disinfection, disinfectant, an antiseptic, a bactericidal agent, a bacteriostatic agent, what is an antibiotic, what is the spread plate technique, and what is the zone of inhibition. Any question about what we're going to be doing and covering in this lab? All right. So as an introduction, sterilization is the elimination of all viable organisms, including any endospores. How we do this in the microbiology lab is usually we stick things in an autoclave, which is sort of like a giant pressure cooker. Disinfection is the destruction of vegetative cells, but not necessarily endospores. Most disinfectants is a chemical agent that's applied to an inanimate object. So chlorine bleach, when you're using it to clean the toilet, it is a disinfectant. An antiseptic is a chemical agent applied to living tissue. Tincture of iodine is a common antiseptic. But let's say I had a cat and it got in a fight and had a really torn up leg, and the only thing I had to clean it, sterilize it, or not really sterilize it, but clean the wound, would be bleach. And I put bleach on the cat. Of course, the cat wouldn't like me for that, but it would clean the cat's wound. I would be using bleach then as a disinfectant. So it doesn't matter what the chemical agent is, it's how it is used. Bleach used on a toilet, an inanimate object, is a disinfectant. Bleach used on a kitty would be an antiseptic. And if you don't know, at least the old method for treating necrotizing fasciitis was to put on gauze on the affected area, and the gauze would be treated in bleach. You'd obviously give antibiotics as well, but that was at least the old way of treating necrotizing fasciitis. So we're using there bleach as an antiseptic as well. Any question about any of that? All right. To evaluate the effectiveness of chemical agents, the surface of a nutrient agar plate is inoculated with a particular bacterium, and you spread the bacteria all over the plate. Paper disc, then saturated with a different chemical agent, is applied to the surface of the agar. The chemical agent will diffuse into the agar, establishing a concentration gradient around the disc. You then incubate the plate overnight and then look at the growth pattern around the disc after overnight incubation. If you have a zone of inhibition, an area with no bacterial growth around the disc, the chemical is assumed to be effective for that particular bacterium. Conversely, if there's no zone of inhibition, the chemical is assumed to be ineffective. Note I'm saying effective and ineffective. I'm not saying sensitive to and resistant to. Any question about any of that? Table 6.1 is listing the different chemical agents, like in alcohol, ethanol, isopropanol, the general action. They are weakly protein denature and lipid dissolution killing cells, either by dissolving the cell membrane or denaturing proteins. We tend to use them as a degerming agent because they are a weak disinfectant or antiseptic. So we use them to degerm the skin, and they do not kill all microbes on the skin, but they reduce the number to a safe level, and then you can inject the skin where you've applied the alcohol. And that's why it's called the degerming agent. And it's because they're weak antiseptics. They're not really a true antiseptic, they're a degerming agent. Aldehydes like formaldehyde, formalin, denature proteins, they are disinfectants. These are unusual disinfectants that these disinfectants can actually kill endospores. Most disinfectants do not kill endospores. Like halogen, tincture of iodine, even chlorine bleach is tincture of iodine used as an antiseptic. It doesn't kill endospores. Chlorine bleach used as a disinfectant most of the time, like when you use it on your toilet, it does not kill at least 50% of endospores, depending on the species. The point is that even chlorine bleach, a very strong disinfectant, does not kill all endospores. Heavy metals used as a disinfectant, occasionally as an antiseptic. Their use for a long time was no longer used because they are toxic to humans. So for a while there, the medical industry was not using them. However, because of the resistance to antibiotics, the use of heavy metals has come back. And we frequently will put heavy metals like in a patient's dressing, like when they have a wound and you don't want an infection to occur in that wound, you can put heavy metals in the dressing or the gauze and then put that on top of the wound. And that heavy metal can prevent the growth of bacteria, including drug-resistant bacteria, meaning antibiotic-resistant bacteria. Peroxygens, like hydrogen peroxide, is used typically as an antiseptic. However, this is one antiseptic that is actually effective at killing endospores. as hydrogen peroxide or peroxygens. Any question about any of that? Phenolics. Phenol was actually the first chemical that Joseph Lister used as a sterilizing agent, not sterilizing agent, antiseptic on patients about to undergo surgery. Phenol was the strongest disinfectant and antiseptic that Joseph Lister found. Nowadays we have many times better agents, but when he was using it, phenol was the strongest antiseptic that he found. They can be used as antiseptics in low concentration. However, the phenolics are irritating to the skin and they can degrade, like this metal surgery instruments. They can also, like phenol, I don't know about some of these other phenolics, but at least phenol is a cancer causing agent. So you really wouldn't want to use that on a patient. Phenolics are typically used as disinfectants, although they are kind of smelly and they're disagreeable odor, but they are good disinfectants. And then surface active agents like quaternary ammonium compounds, they're used as disinfectants, especially in hospitals. Any question about any of this table? All right. So the zone of inhibition size depends only partially upon the sensitivity of the organism to the disinfectant or the antiseptic. Various other factors affect the size of the zone of inhibition, including the concentration of the chemical. The more concentrated the chemical, generally the larger the zone of inhibition. The molecular weight of the agent. The larger the molecular weight of the agent, the smaller the size of the zone of inhibition. And then the density of the bacterial inoculum. The more dense the bacteria inoculum, the smaller the size of the zone of inhibition. So therefore, with the size of the zone of inhibition, you have to control these various variables by standardizing them, or else you can't make a conclusion from the zone of inhibition. Any question about any of that? So when we do the diffusion method, also called the Kirby-Bayer method, after the two people who invented it, we have to standardize a bunch of variables, including all of these three, so that we can then compare the results from one test to another, as well as one operator to another, as well as one person on one place on the planet to another person on another place on the planet. The evaluation of antibiotic effectiveness. An antibiotic can be categorized as bactericidal if it kills the susceptible bacteria, or bacteriostatic if it inhibits the growth of bacteria. If it's only bacteriostatic, what we usually rely on is the immune system to assist in the elimination of the infection, as the bacteriostatic agent itself will not fully remove the agent. It'll just prevent the agent from growing. And then you rely on the immune system to remove the agent. On the other hand, a bactericidal antibiotic will kill the bacteria, assuming the bacteria is susceptible to it. Any question about any of that? The Kirby-Bayer or KB diffusion method is the most common method used for testing antibiotic susceptibility resistance results. So if somebody wants to know if a bacterial infection, we're talking about a doctor wants to know which antibiotic to use, he can take the patient's bacteria causing the infection, spread it over an agar plate, spread the bacteria everywhere so the bacteria will grow in a lawn of agar. And that's why the bacteria have, let me blow this up a little. The bacteria are growing on a lawn of bacteria. Why it looks like white growing almost everywhere except the zone of inhibitions. And then you place on the plate various paper discs with different antibiotics, incubate the plate, and then take a look at the zone of inhibition to determine which antibiotic to use against this bacterial infection. And so you can use the Kirby-Bayer method to determine which antibiotic to use. The filter discs are impregnated with a specific concentration of the antibiotic. You apply the antibiotics and then let it sit for about 5 minutes. And that's mostly to let the agar absorb the liquid when you're putting the bacteria on the plate. And then you place the paper disc down. It has to be placed down at a certain time after you put the antibiotics, generally about 15 minutes, between 5 and 15 minutes after you put the bacteria on the plate. You have to put the antibiotic discs a certain distance apart from each other, and then you have to incubate the plate in the incubator after it's sat out no longer than 15 minutes outside at room temperature, and then place it in the incubator. You incubate it for 18 hours at 35 degrees C. Then you take the plate out and look at the zone of inhibition. You need to measure the zone of inhibition. If there's a zone of inhibition, the antibiotic is high enough to inhibit bacterial growth, maybe even kill bacterial growth, but at least inhibit it. But realize that the size of the zone of inhibition is dependent on several factors. It's dependent on the diffusion rate of the antibiotic. A smaller antibiotic will diffuse further than a larger antibiotic. The degree of sensitivity of the microorganism, the more sensitive the bacteria is to the antibiotic, the greater the zone of inhibition. The less sensitive, the smaller the zone of inhibition. And obviously that one is not sensitive at all because there is no zone of inhibition. It's also dependent on the growth rate of the bacterium and the concentration of the bacteria when you put it in the inoculant on the plate. Let's see, the higher the concentration of bacteria when you first inoculated the plate, the smaller the zone of inhibition. And the growth rate of the bacterium, I believe the faster the bacteria grows, the larger the zone of inhibition. So you have to standardize all of these variables when performing the Kirby-Bayer disc diffusion method, and it's also called simply the disc diffusion method when we're looking at antibiotic susceptibility test. And so here's standardized protocol for applying the Kirby-Bayer disc diffusion method. You first inoculate a broth using several bacterial colonies where the colonies were grown on non-selective media, and the colonies are no older than 24 hours old. Also, you have the bacteria, the nutrient broth, growing no longer than 24 hours. You then measure the turbidity in the broth, and you change the turbidity to 0.5 McFarland units. That's approximately 1 × 10⁵ bacteria cells per mil. And then you put that on your plates. You inoculate only Mueller-Hinton agar plates because different media can affect the size of the zone of inhibition. So we only use Mueller-Hinton agar plates, and the plates are always pH between 7.2 to 7.4 pH. The depth of the agar is always 4 millimeters. If you have deeper agar, the antibiotic will diffuse less in the agar. And then if you have less agar, less than 4 millimeters, the antibiotics will diffuse further. So you always have to use Mueller-Hinton plates, pH 7.2 to 7.4, the depth 4 millimeters, and we always use plates of a diameter of 150 millimeters. You put the bacteria using a non-toxic swab at 0.5 McFarlane units, spread the plate to obtain confluent growth. I'll explain that when I'm going through the lab in a bit, but it literally means you spread it on the plate, you rotate the plate, you do that 3 times. You then place the disc on the plates no more longer than 15 minutes after you put the bacteria down, and no earlier than 5 minutes after putting the bacteria down. You put the paper disc on the plate, and you keep the paper disc at least 24 millimeters apart, no more than 12 discs on a 150 millimeter plate. You then incubate the plates in a non-CO2 incubator for 16 to 18 hours at 35 degrees Celsius. And you put the plates in the incubator no longer than 15 minutes after adding the antibiotic disc. Any question about that? I'm not sure what the CO2 incubator would do to the size of the zone of inhibition. It might not do anything, but we always use a non-CO2 incubator. You always allow the bacteria to grow for 16 to 18 hours. If you allow longer, the size of the zone of inhibition may be smaller. And you always grow it at 35 degrees C. If you grow it at a different temperature, the size of the zone of inhibition could differ depending on what the properties of the bacteria were. If it's growing best at 37 degrees and you grow it at 35 and you put it in at 37 degrees, the size of the zone of inhibition, I believe, could be a little larger. So you standardize the variables by doing this procedure. And then after incubation, you measure the sizes of the inhibition zones in diameter using millimeters. And then you compare the diameter of the zone of inhibition to a table set up by the National Committee for Clinical Laboratory Studies, or the NCCLS, because they set the standards for interpreting whether the zone size is sensitive to that antibiotic or resistant to the antibiotic for a particular one antibiotic, meaning the tables that they generate will have one antibiotic for one concentration, and sometimes they'll have 2 or 3 different concentrations of antibiotics. And then they'll have a size for the zone of inhibition to be sensitive and the size of the zone of inhibition for the bacteria to be resistant. If it's smaller to or equal to the resistant size, that bacteria is considered to be resistant. If it is equal to or larger than the size to be sensitive, then the bacteria is sensitive to that antibiotic. And yes, the size can be between sensitive and intermediate, sensitive and resistant, and that we call intermediate. Going through an example, we're looking at the antibiotic penicillin G. It doesn't have G there, but trust me, that's what it means. We're looking at one organism, Staphylococcus aureus. The size of the zone of inhibition after doing the Kirby-Bayer method, also called the deuce diffusion method, with the protocol stated above, is 20 millimeters for the size of the zone of inhibition. And usually you take that size on two diameters and take the average. And if the zone of inhibition is 20 millimeters, we then look at the NCCLS standards, and we find that if the zone of inhibition is 20 millimeters or less, we say that this bacteria, Staphylococcus aureus, is resistant to this antibiotic. Any question about that? And each bacteria will have a different size for a size being resistant, intermediate, and sensitive for each different antibiotic in the table. So something like Staphylococcus aureus will have a different resistance size than something like Micrococcus luteus to this antibiotic. Any question about any of that? And that's how you determine if a bacteria is sensitive or resistant to a antibiotic. You have to go through the exact Kirby-Bayer method detailed here. Measure the size of the zone of inhibition in its diameter, compare that to the NCCLS standards, and then you can determine whether this species of bacteria is resistant or sensitive to this antibiotic. Any question about any of that? All right, know the terms of this lab. Don't need to do the references. You're not going to be physically performing any of these activities because everything we do will be done online, but you will be held responsible for how to interpret the effectiveness of antibiotics using the Kirby-Bayer diffusion method. In procedure 1, there's a video clip to watch on preparing a spread plate. Review the steps for using aseptic technique when preparing a spread plate or a bacterial lawn of growth. You go ahead and go through that now. Can you guys see this? I don't actually have a plate here, so I'm going to pretend that this is a plate of agar, and I've taken off the lid here. What you do is you take off the lid, and then you dip your swab, pretend this is a swab, and you put it in a bacteria solution that has 0.5 McFarland units of bacteria in it. And you get the Q-tip wet in the I have one of these which is dirty. Yes, this one is. All right. So you dip it in the swab, in the culture, and then you get the worst of the liquid off on the inside of the tube. Remember, the inside of the tube is sterile except for the one bacteria species we're growing in the tube. So you touch the sides of the tube to get the worst of the liquid off. Then you cap the tube, and then you take the swab and put it on the agar plate, and you swab everywhere along the plate. The idea is to get the bacteria growing in a lawn, meaning it grows everywhere. And after you're done, you then rotate the swab a quarter of a turn. You rotate the plate a quarter of a turn. I got to hold it this way now. And then you re-swab the plate. Swabbing everywhere. I don't know if you can see that. Trying to swab everywhere. Can you guys see that? All right, I'm going to assume you can because you can't hear it. Then you rotate.
No, We can barely see it. Your camera's kind of pointed more upward. There's more space above your head than there is. There you go. Now you're more
Let her show.
centered.
All right, there we go.
Yeah.
All right, you'll see it this time. Then you rotate the swab a quarter of a turn. You rotate the plate a quarter of a turn, and then you swab the plate everywhere. You make sure you're swabbing everywhere so you get bacteria to grow everywhere. You then take the swab and you go around the edge of the plate to make sure the edge will have growth on it. Because if you notice, occasionally on the edge, I might not touch the exact edge. So you take the swab and go around the edge. So you do that at least 3 times. And then we say that the plate has a swab of bacteria on it. So it'll grow as a lawn of bacteria, and then bacteria will grow everywhere. You then put the paper disc on the plate. After 5 minutes, you allow the liquid on the plate to be absorbed into the agar for 5 minutes. But you put the paper disc on between 5 minutes and 15 minutes after you put the bacteria on the plate. You then cover the plate, write your initials on it, and after you put the paper disc with the antibiotic on, you put the plate in the incubator no longer than 15 minutes after you put the paper disc on. Usually you put the plate in upside down and incubate it overnight for 16 to 18 hours for 35 degrees Celsius. Make sure I got the time right there. Yep, 16 to 18 hours. We usually do overnight incubation because in the lab, we can't do exactly 16 to 18 hours. So we do closer to 24 hours, which is good enough to do the Kirby Bayer method for the lab. But if you were to do it in a real microbiology lab, you'd want to do it only for 16 to 18 hours. Any questions about that? And so this is giving you the procedure for if you were really doing it in the lab at Clark College, which would be really 16 to 18 hours. All right, in procedure 2, you're going to be evaluating antibiotic effectiveness. You can view the following video clips to complete the virtual lab. So we have a link for preparing a culture for the Kirby biodiffusion method. So you can click that link. And then we have interpreting the results of the Kirby biodiffusion method. So we have a link for that. So click that link. And then we have a virtual lab. Let me see if this works. I say I have a new link, so I might have to get it from the worksheet. But let's see if this works. Doesn't seem to be. There it goes. It's taking its time to load. Hopefully yours will be faster than mine. I'm running Zoom here. And then we have the Kirby biodiffusion method. You want to probably read the description. I'm just going to do the start here. When you go start, it'll show you the first plate, which is after overnight incubation. And we have a bacteria growing on a lawn, although there's no lawn there. Somebody did something wrong with the plate there. They didn't do around the edge of the plate, I'm missing. But we have the lawn of bacteria and the paper disc containing the antibiotic. And as you can see, erythromycin E has no zone of inhibition. Same with P for probably penicillin G. As for streptomycin, has a zone of inhibition. Let's go ahead and you take the ruler, bring it to the edge here, about there. So it looks like the diameter is around 18 millimeters, maybe 17. And so click here, and the correct method, 17, will be greater than 15. And that's correct. If it's green, it's the right color. If it's wrong, it'll give you a red X. It's erythromycin. It's less than 7. We're going to take it wrong. Take it wrong here too. And so that would be yes, correct, less than 13. And then tetracycline, shown here, that looks like about 18 millimeters, maybe 17 millimeters, which is right here. And I got it right, so that's correct. So tetracycline is intermediate. Erythromycin is resistant. And streptomycin is actually sensitive. So that there, which is kind of small, is sensitive. And then tetracycline is intermediate. Any questions about how to do this lab? All right, so that's the first plate. To get the next plate, you have to click the menu button and hit restart. You notice how that's moved to a second bacteria. There's the bacteria there, Pseudomonas aeruginosa. There's the date. It just isn't labeled correctly. It should also have the media here, although we know on this one that the media is always supposed to be Mueller-Hinton agar plates because that's the only one you use. And then you can see that there's a few which have, let's do this one again. It's a little big. I can't get it in the exact place there, close to there. So that one is about 19, maybe 20. So greater than 15, that's sensitive. And then tetracycline will obviously be resistant because it's less than 14. And then how you get the third one, hit the menu button again, restart. You'll have Staphylococcus aureus. And this one you can see is actually multi-drug resistant Staphylococcus aureus because it is resistant to every antibiotic except for vancomycin. If you don't know, vancomycin is the fallback antibiotic we use for treating Staphylococcus or multi-drug resistant Staphylococcus aureus, also called MRSA, and some people use the acronym MRSA. And this one is about 16 millimeters for vancomycin, greater than 12, so it is sensitive. And all the others, like streptomycin, less than 7, will be resistant. Any question about that? And obviously all of these are resistant because there's no zone of inhibition. This right here is showing you the table for the NCCLS, and just says the interpretative standards. The standards are from the NCCLS table, and this column there, I think it's stated, yeah, it's right there, is resistant. This one is intermediate, and that one is sensitive. Any question about that? So that's how you do it. You'll need to do this to answer some of the questions in the lab, by the way. All right, let's go to the worksheet now. So the first question is 2, using the standards required for the Kirby Bayer susceptibility test, an E. coli culture is prepared using the spread plate technique. Antibiotic discs are placed on the surface of the agar and incubated overnight at 35 degrees Celsius. Below are the results with the NCCLS zone standards used for interpretation. So there is the size of the zone of inhibition for ampicillin, cephalothin, gentamicin, and tetracycline. And here we have the NCCLS standards. We're only looking at E. coli here. We're not looking at Staphylococcus aureus. And for ampicillin, I believe I've got the results for you in the table here. For ampicillin, the size of the zone of inhibition, you measure it here. Taking a look here, you see that the size of the zone of inhibition is here, which is 15 millimeters, which is there on the table. And the interpretation for this one, I'll give you the first one. For 15, it means for ampicillin that it's intermediate. Any question about that? Now, how you measure the size of the zone of inhibition is you do not measure from here to there, which would be about 9 millimeters, maybe 9.5, 10. Because if you notice, the 0 is not here. So you don't measure from there to there because the measurement starts from 0. And so you go from 0 to 0 to get your measurement of the diameter. So from here, on cephalothin, you measure from here to there to get your measurement of the zone of inhibition. Once again, you do not measure from here to there. So if you go 9 millimeters, it'd be wrong. Any question about any of that? And that's just the way calipers work. So take the measurement of the zone of inhibition and put it in the table here, and then give your interpretation, put it here. I've done the first one for you. All right, that's question 2. One is just going through the, looking at the video, the 2 videos in the virtual lab. For question 3, then list 5 variables that must be standardized when performing an antibiotic susceptibility test using the Kirby-Bayer disk diffusion method. And if you remember, we went over that in a table as well as in the text of the lab module. 4, the size of the zone of inhibition often varies for 2 different species of bacteria when challenged with the same antibiotic. What is a possible explanation for these differences? Give your answer here. 5, what would happen if we didn't use aseptic technique to put antibiotic disc on the plate? And then also mention what will happen to our test results. So if you don't use aseptic technique and you put your antibiotic disc on the plate, what's going to happen to the plate? And then what will happen to our test results? All right, 6, what would happen if we increased or decreased the concentration of the antibiotic in the disc? Be specific in your answer, meaning you have to tell me both what will happen if you increase the concentration of antibiotic in the disc, and then tell me also what will happen if you decrease the concentration of the antibiotic in the disc. And I'm only looking for the size of the zone of inhibition. You don't have to tell me about whether or not this will affect the NCCLS table. People are actually beginning to tell me that, and I'm assuming they're putting this into an artificial intelligence, and it's giving you that answer. And if you do that, remember, you're supposed to tell me in in text where you got your answer from. So if you use AI, you should be telling me where you got your answer from. And what I should do is dock people if they don't tell me what the AI is, because I know that if you're telling me an answer like that, you didn't get that on your own. You put it in and got it from AI. But just tell me what will happen to the size of the zone of inhibition. And 7, antibodies can be classified as bactericidal. They actually kill the microbes. For example, penicillin G is actually a bactericidal antibiotic. Well, it kills the microbes. Or if the antibiotic is bacteriostatic, it inhibits the microbial growth but does not kill the microbes. So when you're observing a zone of inhibition around a disc, how would you determine whether the antibiotic causing that zone was bactericidal or bacteriostatic? This is a thinking question. So I'm going to warn you that a lot of you will get this question wrong on your first attempt. But most students do get this question right on their second attempt. So this is a thinking question. It's not an easy question. You won't find the answer directly in the lab module. You have to think about it. Any questions about number 7? All right. 8.
So really, I do have a quick question on that one for 7. When we're looking at the actual zone of inhibition, we're not looking at the inside, like the actual cell itself, correct? We're just comparing the zone of inhibition.
Even if you looked at the actual cells, like under a microscope, how would you know if that cell was killed or not? You can't tell under the light microscope. Okay, so I guess you could
Okay.
use an electron microscope. You might be able to answer the question, is the cell alive or dead with the electron microscope? Although you have to look at the entire boundary of the cell probably to answer that question. But with the light microscope, you would not be able to tell if the cell was alive or dead. And I'm not even going to address the electron microscope because with the electron microscope, you would not know if the cell was recently killed or if it was alive. You may not.
Okay, so when we're looking, we really just need to look at the zone on the disc.
Yeah, I'm asking you only to look at the disc with your eyes, and how can you tell? Okay,
Okay.
what can you do to answer this question? You might have to do something more than just look at the disc. Okay, all right. A, when performing antibiotic sensitivity testing, it's important that the test conditions are kept very standardized. We use the same kind of media, Mueller-Hinton. We always use the same agar depth, 4 millimeters. We use the same age of the culture. Why do we standardize all these variables? So answer that. And that one actually is given to you in the lab module. 9. Are there any other substances besides antibiotics we could evaluate using this disc diffusion method, also called the Kirby-Bayer method? Name a substance. So you can say yes, that is a correct answer, or no, that is a correct answer, but you have to name one substance. Any question about any of that? All right, 10. Does the presence of a zone of inhibition necessarily mean that the bacterium being tested is sensitive to the antibiotic? Explain your answer. 11. From the size of the zone of inhibition on chloramphenicol, abbreviated C, on E. coli in the virtual lab, is the bacteria sensitive, intermediate, or resistant to that antibiotic? For chloramphenicol on Pseudomonas aeruginosa, P. aeruginosa in the plate, is the bacteria sensitive, intermediate, or resistant to the antibiotic chloramphenicol? Any questions about question number 11 or any other question? All right, if there's no questions, I'll be here until 8 o'clock or the last student logs out to help you with any questions. If you do have a question, make sure you ask it before the last student logs out, because once the last student logs out, I'm going to log out as well. But I will be here until 8 o'clock or until the last student logs out. Any question? All right, get started.